Assalamu alaikum dear students. I am Dr. Shahjabeen and today's lecture topic is regulation of respiration, which is a complex integrated control mechanism. It can be modified by some respiratory reflexes and chemical control mechanism. This lecture is concerned with neurogenic system for regulation of respiration. These are the objectives of this lecture. At the end of the lecture, you would be able to name the centers for the neural control of respiration and briefly describe their functions. Describe the location and function of the medullary rhythmicity area and their role in normal quiet breathing and forceful breathing. You would be able to identify the role of Pneumotaxic and Apneustic Centre in the Pontine for the control of respiration. Neural mechanism basically comprises of automatic control system of involuntary neurons that are present in medulla and pons, along with the afferent impulses to these respiratory centers that can change their activity according to the body's demand or needs. Automatic control system is the involuntary neural control system that regulates respiration by several group of neurons situated bilaterally in the medulla and pons in the brain stem. Here you can see in the picture some neurons are present in the medulla, these blue colored, and these group of neurons are known as medullary respiratory center, while the group of neurons that are present in pons are known as pons respiratory centers. Together these are known as respiratory control centers in the brain stem, and they are involuntary. Here you can see a pre-Botzinger complex. This is the area that is present in the medulla and is essential for the generation of respiratory rhythm. A group of neurons or a cluster of neurons that are located in the medulla are known as pre-Botzinger complex. Here you can see in the medulla pre-Botzinger complex. These pre-Botzinger neurons or complex display pacemaker activity. What is pacemaker activity? These are the cells that undergo self-induced action potential similar to those of SA node of the heart. Their function is to discharge and initiate synaptically rhythmic activity and drive the other cluster of neurons, especially the DRG. We will do it in the next coming slides. The dorsal respiratory group of neurons that are present, they are also present in the medulla. In turn, these DRG neurons fire rhythmically and form basic rhythm of respiration. So these cluster of neurons, the pre-Botzinger complex that is located in the medulla, are the respiratory control pattern generator, or you can say these are the medullary rhythmicity area. The respiratory centers that are present in medulla includes two group of neurons. The dorsal respiratory group, it is located in the dorsal portion of the medulla. Here you can see it. This is the picture I have taken from your guidon. And these DRG mainly causes inspiration. Other group of neurons are known as ventral respiratory group or VRG, located in the ventrolateral part of the medulla, which mainly causes expiration. Dorsal respiratory group of neurons plays an most important role in the control of respiration, and most of the neurons are located within the nucleus of Tractus solitarius and some in the adjacent reticular substance. Here you can see medulla, and these are the gray colored are dorsal group of neurons. These are the neurons that control inspiration and the respiratory rhythm, actually that is generated by the pre-Potzinger complex. You know it now, the medullary rhythmicity area. The neurons of TRG are of three types: inspiratory neurons, inspiratory off-switch neurons, and the integrator neurons. Most of the DRG neurons are inspiratory neurons that discharge during inspiration and initiate normal inspiration. They send nerve signals to the muscle of inspiration. That's why these are known as inspiratory neurons. The signals these neurons send are not instantaneous, but in a ramp fashion. What is ramp fashion or ramp manner? Ramp means in a steadily increasing manner. That is, weak in the beginning and then swiftly, slowly increase for about 2 seconds. These signals are called inspiratory ramp signals. Because of these signals, the lung volume increases slowly, steadily, rather than abruptly to make a person gasp. Ramp signals then cease for 3 seconds, next 3 seconds, and then another cycle begins. Inspiratory switch off neurons are the group of neurons that are responsible for terminating the activity of inspiratory neurons and causes turning off of excitation of muscles of inspiration. What are those? Diaphragm and external intercostal muscles. These muscles then relax, and due to the elastic recoil, expiration occur. The integrator neurons integrate the activity of inspiratory neurons and inspiratory switch off neurons. These neurons are located near the inspiratory neurons and are stimulated by these inspiratory neurons. Whenever they are stimulated, they integrate to inspiratory switch off neurons, which in turn responsible for terminating the inspiration or inspiratory ramp. You know their function. They terminate the inspiration. So, what is going on? Inspiratory neurons through integrator neurons indirectly switch off their own inspiratory ramp. This forms the basic circuitry of our automatic respiration rhythm, like inspiration and then expiration. These integrator neurons also receive both the excitatory and inhibitory inputs. They get excitatory input from cerebral cortex, Pneumotaxic Centre, and some of the vagal efferents from the stretch receptors. Whenever these integrator neurons get excitatory input. These stimulate inspiratory switch off neurons and cease ramp signal. While they also get inhibitory input from the Apneustic Centre and some of the input from the Medullary inhibitory center also. And when they get inhibitory signals, they inhibit inspiratory switch off neurons and thus delaying the inspiratory ramp, delaying the switching off of inspiration. This is the pictorial representation of the previous discussion for your better understanding. In the middle, in the yellow color, you can see the integrator neuron that is always stimulated by inspiratory neuron. The inspiratory neuron, whenever stimulate the integrator neuron, it will stimulate the inspiratory switch off neuron, which in turn causes the switching of inspiration or inspiratory ramp. So this is the normal inspiratory and expiratory cycle, our normal respiration. But this is not as simple, as integrator neuron also receive excitatory and inhibitory input. When they get excitatory input from vagal afferent stretch receptors, from the lungs or cortex or pneumotaxic center from the Pontine, they again excite inspiratory switch off neurons, and again it will cause early switch off of inspiratory signal. Then what will happen? Yes, there would be shallow and rapid respiration. And when the integrator neurons are inhibited by Apneustic Centre, look at the minus sign, or sometimes by the medullary inhibitory neurons, they will inhibit inspiratory switch off neurons and so prevent the switching off of inspiratory ramp. In this way, they causes the increased duration of inspiration and increased tidal volume. Ventral respiratory group of neurons are located at the ventrolateral portion of the medulla. You can see it. This one, yellow and red one in the medulla. Ventral respiratory group neurons contain both inspiratory cells called inspiratory neurons and expiratory cells called expiratory neurons. Do remember, DRG, dorsal respiratory group neurons, don't have expiratory neurons. The inspiratory and expiratory neurons have inhibitory connections to each other, or you can say they are reciprocally innervated with each other. For example, if the motor neuron to expiratory muscles are active, the neurons supplying to the inspiratory muscles are inhibited, or vice versa. Ventral respiratory group of neurons remain inactive during quiet breathing. Which group of neurons perform the function of normal and quiet breathing? Yes, DRG, dorsal respiratory group of neurons. But these ventral respiratory group of neurons become active during forceful respiration, as these neurons contribute to both inspiration and expiration, and become active during inspiration through inspiratory neurons as well as during expiration through expiratory neurons. These VRG neurons operates when high levels of pulmonary ventilation is required, as they send powerful, strong expiratory signals to abdominal muscle during heavy exercise. Now come to the Pontine respiratory centers located in the pons of brain stem. Include Apneustic Centre and Pneumotaxic Centre. Here you can see in the pons Apneustic Centre and Pneumotaxic Centre. Both of these centers modify the activity of medullary respiratory centers, DRG and VRG. Apneustic Centre refers to a group of inhibitory neurons that are present in the lower part of the pons. As the name says, apneustic center causes apneustic respiration or breathing or apneusis. What is apneusis? It is abnormal pattern of breathing characterized by deep, full and prolonged inspiration. The mechanism we have already done in the integrator neuronal function, that apneustic center sends inhibitory signals to the integrator neurons of DRG that affect the inspiratory switch of neurons and prevent the switch off of the inspiratory ramp signals from inspiratory neurons of DRG that causes the increased tidal volume and duration of inspiration, resulting in a deeper and more prolonged inspiratory effort that is, I have told you, known as apneusis. Normally, apneustic center is inhibited by vagus nerve and the pneumotaxic center. Pneumotaxic center is located in the pons, in the upper pons. And as I have told you, pneumotaxic center inhibits apneustic center. When the pneumotaxic center is stimulated, it shortens inspiration, leading to shallow and more rapid respiratory pattern. Through the integrator neurons, you know it. And what was the apneustic center doing? Yes, it was making the inspiration deep and prolonged, just the opposite one. Thus, though rhythm of the respiration resides in the DRG neurons in the medulla, pneumotaxic center and apneustic center control these neurons to regulate the depth and rate of respiration or modify the respiration. Now we have done all the respiratory centers that are located in the medulla and pons and their functions too. The activity of the respiratory center is also influenced by afferent impulses from lungs and other parts of the body, like afferent impulses from the higher centers like cortex, neocortex or limbic system. The voluntary control of the breathing can be done like in talking, singing, swimming. In swimming, breath holding or voluntary apnea can also be done by stopping breathing voluntarily for about 50 to 60 seconds. Pain and emotions can also affect rate and depth of the breathing, and breathing can be increased or hyperventilated voluntarily. One of the protective afferent impulse from pulmonary stretch receptors is Herring-Brewer inflation reflex. Stretch receptors are located in the muscular portions of walls of bronchi, bronchioles, and throughout the lungs. And whenever lung is overstretched or whenever tidal volume is increased more than 1 or 1.5 liters per breath, these stretch receptors send signal via the vagus nerve to the dorsal respiratory group of neurons and limit inspiration, just like Pneumotaxic Centre, causing shallow and rapid breathing. So, Herring-Brewer inflation reflex is lung overinflation signal that limit further inspiration and excess lung inflation. A few sensory nerve endings are present in the alveolar wall in juxtaposition to the pulmonary capillaries. Due to their location, these are known as J-receptor. They are sensitive to increase in the content of interstitial fluid between the capillary and the alveolar epithelium here. Therefore, they are stimulated in the pathological conditions like pulmonary congestion, edema, pneumonia, or in congestive heart failure. And in physiological conditions like after heavy exercise, or some of the chemicals also stimulate them like halothane or bradykinin. Whenever these receptors are excited, they give a person a feeling of dyspnea. Other afferent impulses that can affect respiratory centers are irritant receptors. Irritant receptors are located below the mucosa of whole respiratory tract. These are stimulated by a variety of chemical stimuli like serotonin, prostaglandins, bradykinins, ammonia, or simple by smoke. An important function these receptors may perform is to detect pathophysiological processes in the airway, such as chemical irritation, inflammation, or congestion. These receptors also detect histamine that causes bronchoconstriction in asthma and can initiate protective reflexes like cough and sneezing. And you will do these reflexes, cough and sneezing, in detail in the next lecture. Thank you, students. This is my email address for your queries and the reference books names.